Hello, everyone. Welcome back. Thank you for joining us. I'm Steve.
And I'm Scott.
And together we are Backyard Musings. We're broadcasting live. Well, I'm in Apple Valley.
And I'm in Hurricane, Utah.
On assignment. Scott's on assignment. He's doing some Sasquatch research for us.
Yeah, melted Sasquatch. So, yeah, but we are talking science and technology today.
And we are not the experts.
No.
We're the messengers.
Absolutely not.
Okay. A new study reveals that blocking ribosomal RNA production rewires cancer cell behavior and could help treat genetically unstable tumors. Researchers at Johns Hopkins Kimmel Cancer Center and the Department of Radiation Oncology and Molecular Radiation Sciences have identified a tumor-suppressive response that could lead to new treatments for cancers that are difficult to treat. This study was published June 18, 2025 in Cell Chemical Biology and partially funded by the National Institute of Health. Would you like to know more?
The team showed that interfering with a key step in protein production can inhibit cancer cell growth. The research also explains why certain cancer cells are particularly sensitive to this approach. These findings point to the new therapeutic strategies for cancers with common genetic mutations. The team discovered that blocking RNA, polymerase one or Pol1, the enzyme responsible for transcribing human ribosomal RNA, triggers a unique stress response. This response alters RNA splicing, the process by which cells generate different forms of proteins and leads to tumor suppression. Ribosomal RNA genes are crucial for building ribosomes, the cellular machinery responsible for transplanting proteins. Okay, I did not know that. Never heard any of that.
Here's a quote. Ribosome biogenesis has long been known as a hallmark of cancer, says study leader Mariki Laiho, medical doctor and a PhD. The Willard and Lillian Hackerman Professor of Radiation Oncology and the Vice Chair for Research of the Department of Radiation Oncology and Molecular Radiation Scientists. The quote continues, our study reveals that the ribosomal protein RPL22, typically a structural component of the ribosome, plays an unexpected dual role as a critical regulator of RNA splicing.
In 2014, Mariki Laiho and her team identified RNA polymerase 1, or Pol1, let's just call it Pol1, as a promising therapeutic target for cancer treatment. She began laboratory research using human cell lines to test a small molecule called BMH21. This compound was developed in collaboration with James Barrow, PhD, a pharmacology and molecule sciences expert at Johns Hopkins, to block Pol1 activity. Okay.
Yeah, so he's an expert.
I guess, yeah. Let's hope so.
Okay.
Messing with pretty cool stuff.
Yeah. In their most recent study, the team examined more than 300 cancer cell lines and found that tumors with mutations in the gene RPL22 or with elevated levels of MDM4 and RPL22L1 were especially responsive to Pol1 inhibitors like BMH21, whatever that means, and a newly developed drug called BOB42. These genetic changes are frequently found in cancers with mismatch repair deficiency, or MMRD, including colorectal, stomach, and uterine cancers. Okay, let me say that again. Colorectal, stomach, and uterine cancers.
Yeah, all nasty cancers.
Yes. MMRD allows copying errors in DNA to go uncorrected during cell division, leading to a high number of mutations and a greater risk of cancer development.
The team tested the Pol1 inhibitor BOB42 or BOB42 in animal models, including patient-derived tumors containing the same key genetic markers. The drug reduced tumor growth by up to 77% in melanoma and colorectal cancers. That's pretty good. Yeah, that's good.
Quote, these findings highlight a promising new path for targeting cancers, especially for patients with mismatch repair deficient cancers that are resistant to existing therapy, says the study's first author, Wenjun Fan, a PhD research associate.
The study also suggests that changing how cancer cells splice RNA or produce different forms of proteins could affect how the immune system recognizes tumors. Combining immunotherapies with PAL1 inhibitors may improve the effectiveness of immunotherapies.
Quote,
Okay.
this is an entirely new conceptual framework for understanding how rRNA synthesis influences cancer cell behavior, says Laiho. Targeting this pathway could not only suppress tumor growth, but also modulate tumor and antigenicity and enhance responsiveness to immunotherapies. I
Okay,
think this is promising. I mean,
they've had some good results. 77%. That's a huge number.
going in the right direction. I mean, if you have a loved one or you have these kind of cancers, a lot of men have rectal cancer and a lot of women have the uterine cancer. So
Yeah,
this is good news.
Yeah. Yeah, and that 77%, that's almost as high as Trump's approval record rating right now.
Almost.
Yeah. Okay. Good one.
All right, folks. Thanks for joining us. We'll catch you on the next one.
Take care, everyone. Thanks.